This is for number 9. So in pea plants, tall is dominant to short, and yellow seeds is dominant to green seeds. Show the expected phenotype ratio in the offspring for a genetic cross between homozygous tall, homozygous yellow seed producing plant. So that means the genotype of the first parent is big T, big Y, big Y. Homozygous for both dominant traits. With a short green seed producing plant. So that means the second parent is homozygous recessive for both. So we've completed A, the genotypes of both parents. B says perform a Punnett square to determine the expected phenotype in F1 generation. If you do the FOIL, you will notice that you can only produce one type of gamete in either parent. So 100% of the F1 will be double heterozygous. But phenotypically, 100% tall and yellow seed producing, right? Okay, moving on to part C. 2 of F1 were crossed together to produce F2. Perform a Punnett square analysis to determine the expected phenotype. Now, we're crossing 2 double heterozygotes with one another. Okay, now you can do this 4x4 Punnett square by figuring out the 4 possible gamete produced from either parent. However, this is a special case where we can use the golden ratio, right? So the golden ratio is the ratio that we can use only when we're crossing 2 parents that are double heterozygotes. Meaning both parents need to be heterozygous for both genes. And that golden ratio was 9 to 3 to 3 to 1. Which means 9 out of 16 offspring will have dominant traits for both. 3 out of 16 will have 1 dominant, 1 recessive trait. Another 3 out of 16 will have 1 recessive, 1 dominant trait. And finally, only 1 out of 16 will have 2 recessive traits. So that's what we expect from, even without doing the whole Punnett square. Moving on to part D here. The following phenotype is observed in F2 generation. Perform a chi-square analysis to determine if the actual results correlate with the expected results. So these are our observed results, and now we're going to make a separate column for expected results. So what we first need to do is we have to figure out the total number of offspring by adding up these 4 numbers. So I'm adding up these 4 numbers, and the total comes out to be 800. It's a pretty number. Now, out of 800, 9 over 16 will have tall yellow seeds. 3 out of 16 will have tall green seeds. 3 out of 16 will have short yellow seeds, and only 1 out of 16 will have short green seeds. So I'm going to take 800 and multiply 800 by each of these fractions to figure out the expected numbers. So they come out to be these numbers. Again, these numbers should also add up to 800. Okay, so now let's do the chi-square calculation. The chi-square formula goes observed minus expected squared divided expected summation. So you're going to set up the chi-square calculation for all 4 of these rows here. So I'm going to do that really quickly. And now you should put this into your calculator. Okay, I got 7.72 as my chi-square value. And since there are 4 possible outcomes, my degree of freedom is 3. And critical value is going to be, let me see. Let me look on the very last page. Okay, degree of freedom. In the 0.05 column, the critical value is 7.81. So let me go back to our question. So critical value is 7.81. So since our chi-square value is similar, but it's still smaller than critical value, we say we fail to reject, and therefore the expected result and the observed results are correlating with one another. So that's the overview of number 9. So that means that these two genes assort independently. The gene determining the plant height and the gene determining the seed color, they are two genes that assort independently, meaning they're located on separate chromosomes and they are not affecting each other. So that's the overview of number 9.